Hello everyone, welcome back to my channels. Today I would like to show you how to perform immunohistochemistry or immunofluorescence staining using an adherent cells.
You only live once. Educate your life. Welcome to the Learning Factory by Abdul Malik Setiaw.
So first of all, I would like to explain how I do my immunofluorescence staining. So first, I'm using a stem cells, mesenchymal stem cells. Mesenchymal stem cells are adherent cells that will attach to the culture plate. So we have to detach the cells first using whatever methods that your lab is doing. But for me, I'm using a TE 0.05 TE to detach the cells, and then after that I spin the cells and suspend the cells in 2% FBS, 2% FBS in PBS. So after that, I took 100 until 200 microliter per slide and put it inside the cytofunnel. So the cytofunnel, then we put into the cytospin machine, and we can spin the cells using 1000 until 1500 RPM for 5 minutes. So this method allows you to have smaller number of cells to stain. This method, in comparison with growing the cells in a cover slip that require a larger surface area, and you also need more reagents to stain. So if you don't have many antibody, primary, secondary antibody, or even DAPI, you can use this method. So later on, you might only need, let's say, only 50 to 60 microliter of antibody, primary and secondary, per slide. So it's quite efficient. So after we spin the slides, we just wait for the slide to dry up, maybe for 30 to 1 minute, 30 seconds to 1 minute in room temperature, and then we straight to fix the cells using 4% of formaldehyde. So you can use the 4% of PFA, and we put around 100 microliter to each slides, which is quite small. The surface, the cytospin allow you to spread the cells in a very small surface area, around maybe half of centimeter of diameter. So we incubate using 4% PFA for 10 minutes only, and then after that we have to wash with PBS or washing buffer. You can use PBS only, or you can add Triton as well. But if you don't add a Triton, eventually you will going to permeabilize the cells using 0.5% Triton X100 in PBS for 15 minutes. So this permeabilization method will destruct the cell membrane. So later we can incubate the antibody, and the antibody can enter inside the cells. After that, we then wash the Triton using washing buffer, and we then block the non-specific binding of the cells using 5% of goat serum. You can use BSA as well, or maybe FBS, 10% of BS, but the best method is using 5% of goat serum. And if you using secondary antibody from goat as well, so it might be better if you use 5% of goat serum to block this non-specific binding. We block the slide or the cells for 1 hour, and then we don't wash after that. We just discard or remove the blocking buffer, just flick the slide, and then we add the primary antibody. For me, I'm using anti-CK3 and anti-octomorph. You can do a single primary antibody or double primary antibody. It's up to you. If you need more than one, you can just do you can just mix 2 primary antibody from different species. For me, I'm using mouse and rabbit. You incubate the primary antibody in at 4 degrees Celsius overnight, or you can do it around 3 hours at room temperature, but the optimum condition should be on 4 degrees Celsius overnight. After that, you in the following days, you just remove the primary antibody and then wash with PBS or washing buffer, and then you can add the secondary antibody and washing it again, and then add DAPI. Okay, so but if you use a double primary antibody, so you have to mix those secondary antibody together, and then just add the secondary antibody first, and then incubate for 1 hour, and then wash again, and then after that you add the DAPI for 1 hour. So you basically finish the staining, and then you add mounting agent, and then you cover the slide with cover slip, and you just keep it the slide inside the box and keep it at 4 degrees Celsius, and you ready to see the cells in the next day or whatever you want. Just keep it the box keep it inside a dark box, so the fluorescence will not decrease. Okay, so this is the cytofunnel. Cytofunnel is some device that allow you to dispense or spread the cells using the cytospin. So you just put 100 microliter or maybe 200 microliter of cell suspension, and you just inside the cytofunnel with the slides attached, the object glass attached, and then you just put this combination inside the cytospin and spin it for 1500 rpm around 5 minutes. It's enough. And this is the method that I use to incubate the primary antibody and the secondary antibody without using PAP pen. So it's better for you to use the PAP pen, but if you cannot afford the PAP pen, I'm using this proximal part of the tip, or 1 mil tip. I cut around maybe 3 millimeter, and then I just add a Vaseline to it, so it will block the antibody solution and keep the antibody solution inside the circle, so the antibody will not getting everywhere. So that's why if you only have like small amount of primary and secondary antibody, you can use this method, and I've already tried the method and it's actually perfect. You don't have to be worried about the Vaseline, because eventually we just wash them with washing buffer, which contain the triton, which is basically detergent. Okay, so this is the method that I did to stain the E8 immunohistochemistry and immunofluorescence staining, and this is the result. This is the DAPI, and the green one is the octomorph, and the red one is the CK3. We just collect this image through the microscope, fluorescent microscope, and then this is the merged image of 3 of those color. Okay, thank you for watching. Hopefully you will gain more confident to do the immunofluorescence staining. Okay, bye.
People do many things in their life. Sometimes we fail, sometimes we succeed, but we always learn from them. Welcome to the Learning Factory by Abdul Malik Satya.